I'll let you take it from here.
Thank you, Amy. Thank you so much for that introduction and for inviting me to be here today. I'm really glad to be back in Seattle and back at SAFS. As Amy alluded to, my background is in oceanography and biology, but I've sort of transitioned into more of an ecological statistics role. So that's, I would say now that I'm an ecological statistician. As I was putting together this talk, I realized that actually I work on a lot of different species in different regions using different techniques. And so it was a bit difficult to sort of consolidate that all into one coherent talk. So I wanted to start by laying out some themes that I think recur in the different types of research projects that I work on. The first is that I primarily work on marine mammal population assessment. So I do work on cetaceans, including whales, dolphins, and porpoises, and also on pinnipeds like seals, sea lions, and walrus. All of the work that I do is applied in the sense that it is always motivated by a conservation or management question. This is not theoretical or abstract. There's always sort of a problem that we're trying to solve. And I specialize in quantitative methods development for the purpose of marine mammal population assessment. I particularly am interested in models that make use of imperfect data. I mean, all data is imperfect in some way, but sort of thinking about how we can best use existing data sets and combine them effectively. One way that I do this is through integrated population models. I'm also interested in developing models for new data. So as we develop fancy new machines that generate new types of data, how are we going to deal with those statistically? One example of this is close-kin marker capture. So today I'm going to focus on these two modeling techniques, integrated population models and close-kin marker capture, as examples of ways that we can use quantitative techniques to assess marine mammal populations. I'll start with integrated population models and go over the basic concept of what they are and how they work. I'll compare them to stock assessment models, which the fishery scientists in the room will be more familiar with, I think, and then give some marine mammal examples. So one example from marine mammal examples from my own work, I should say. One example I'll give is on beaked whales, showing the value of simulation studies to improve data collection. And one on beluga whales that actually came out of my postdoc here at SAFS on the past, present, and future of Cook Inlet beluga whales. And then I'll outline a couple of directions of future research that I'm interested in. So the basic concept of an integrated population model is that you have a central process model that is a Leslie-type matrix population model that moves the population forward in time. You then add submodels to account for your different data types, so different observational submodels. That makes this a state space model because you're modeling the process, the population process, and how the population is changing over time, separately from how you're modeling the observations, how those data are collected, and the issues associated with that. Each submodel, each of those observational submodels is linked to the main process through at least one shared parameter. That's what connects this all together. So here is that same idea in graphical form. So I'll walk through this. This is an example from the literature for a bird population. So the central box in the middle represents the population process, where this N is the number of individuals in the population. Those individuals are counted, producing some data Y. Now, we also have some other types of data available for this population, including productivity data, in this case, counts of fledgling birds in nests, and capture-recapture data, so capture histories for some of the birds. So we can link these together because the population process and the number of animals in the population depends on these life history parameters that are shared with the observational submodels. So this is representing the birth rate. So both the productivity data and the counts provide information about that birth rate. And then on this side, the capture histories M, which we acknowledge are observed imperfectly, provide information about adult and, in this case, juvenile survival. IPMs often use a Bayesian implementation. And one of the big advantages of that is that you can incorporate prior information. And I'll give an example of that because I think that I think is really cool from a project I was involved in with harbor seals. So for this project, we had some data specific to that was more recently collected that could inform our estimates of things like survival and fecundity. but we didn't know what historical survival and fecundity looked like for this population. So we looked to the literature and compiled all of the available estimates of vital rates from the literature for harbor seals. So this is for, the example I'm showing is for fecundity. These are a bunch of different references. And then these are the fecundity rates that each of those references reported. And you see that some references report a single point estimate. Some have confidence intervals around that. And some references even separated out fecundity of first-time breeders from fecundity of repeat breeders. So we have all this information, but there's obviously caveats associated with each of those studies. So we compiled basically a literature review and circulated it to a panel of experts in harbor seal biology and asked them to review that literature and give us their best opinion of what fecundity was most likely applicable to the population we were interested in. So that looked like this. So each expert came up with what they thought fecundity should look like based on what was in the literature. And notice that we didn't ask them for a point estimate. We asked them for what do you think is most, we asked them to provide information about what they thought was most plausible that then was converted into a distribution. We averaged those distributions across all 10 of the experts to produce this black line. All of the experts agreed that that seemed reasonable. And so we used it as a prior in our integrated population model. So this allowed us, I think this is really cool because it allows us to borrow from the past and not reinvent, to really look back at all the work that's been done and take what we can from it moving forward in a quantitative way, in a very explicit way. So those of you who are involved in fisheries stock assessment might be thinking that this IPM thing sounds kind of familiar. It's in some ways quite similar to stock assessment models. So I was involved in a project with some collaborators, both fisheries scientists and conservation ecologists, came together to compare IPMs to stock assessment models. And this came out of the Center for the Advancement of Population Assessment Technology. So we tried to see the ways in which they were the same and the ways in which they were different. So integrated population models have typically been developed or historically been developed for terrestrial species, particularly birds, and usually are on quite a small scale, both in terms of the number of animals. So we're talking hundreds of individuals, and small spatial scales, so small little populations in restricted areas. Integrated population models are often happy to estimate relative abundance or trend over time as opposed to absolute abundance. They make use of more individual level data like capture, recapture histories for individual animals. Because they're relatively small scale, small n, it's tractable to fit them in a Bayesian context and to use explicit priors like I just explained. Stock assessment models, on the other hand, come from the aquatic and marine worlds, where people are usually dealing with very large N of hundreds of thousands to millions of animals, or are interested in biomass as opposed to number of individuals. On large spatial scales, usually, because stock assessment models are usually designed for extraction purposes to inform management, they're interested in the absolute number of animals or the absolute biomass. They make use of more population level data just because it isn't possible to collect a lot of data on individual fish, for example. Usually fit the models using maximum likelihood estimation. And then for parameters that can't be fit by the model, they do things like tuning to make the model work. So some similarities and some differences here. And it's more, I think what we found is that these models are influenced both by the objectives. So we acknowledge that stock assessment models have an objective that is often different from integrated population models, but also it's just a tradition of, oh, we've always done it this way, or, oh, my advisor did it this way. So we found that really the methods could borrow from each other and that we could be a little more agnostic in terms of how we decide what kind of model to use. So the paper actually provides some guidance for what to think about when you're thinking about whether to use a stock assessment model or integrated population model, or how you can combine aspects of both of those. And hopefully we can continue to collaborate this and communicate. A lot of the barriers were just language, like we call the same term different things in these different disciplines. So hopefully by increasing communication and collaboration going forward, we can learn from each other and improve both fields. Now I'll present an example of an integrated population model. So this was a simulation study. This was funded by the US Navy because they're interested in how they can best monitor for disturbance of marine mammals around Navy training sites. And it was called Marine Species Monitoring for Population Consequences of Disturbance, but really it was Marine Mammal Species Monitoring for Population Consequences of Disturbance. And the project had three main goals. The first was to look at what data already exist for priority species and regions for the Navy. So what's already out there? What have people already done? Can we put it all together in one database and make better use of it? The second part of the project was to use dynamic energy budget models. And this was to identify potential early warning signs of disturbance in populations. So usually when we think about monitoring for disturbance or detecting disturbance, we think about looking for a change in the population size, but maybe there's something about their life history that changes earlier than that. So for example, maybe if we could look at survival or fecundity, we might be able to detect a decline earlier than if we waited for the actual number of animals to change. Then the third part of the project, which is the part that I was involved in, was evaluating whether integrated models, integrating these different types of data, could improve power to detect trends in abundance. So what data are already collected? I'll give an example for Cuvier's beaked whale in Southern California. These are different modalities of data collection, from visual tag, passive acoustic, capture, recapture, and biopsy. And over about a 20-year period, you see that there's actually quite a lot of data available, not in every year, not for every type, but there's a good amount of data. From the dynamic energy budget modeling, we found that as populations are disturbed, we expect the first thing to go to be the number of calves in the population, or the proportion of calves relative to adults. So basically, if you think about a population that's stressed, or an individual that's stressed, it's the easiest thing to do is to not reproduce. So there's this signal when right after the initial onset of disturbance, and then as the population returns to equilibrium, starts to recover, it rises back up again. So then the third question of whether we could use integrated population models to improve power to detect trends in abundance. This was so like I said, it was a simulation study based on Cuvier's beaked whales in Southern California, but simulation. So what I did was simulate a lot of populations under different possible scenarios of decline or stability in the future, and then simulated different types of data collection, both in terms of types of data collection and frequency. And then I built an integrated population model to analyze those simulated data and evaluated whether we would have been able to detect whatever scenario I had simulated over here. The model looked like this. So similar to the one I presented before, in the center is the population process model, where the total population size N is influenced by life history parameters, including survival rates, carrying capacity, and fecundity. Now, the three of the data types that exist for this population already are photographic capture, recapture, passive acoustic surveys, and visual line transect surveys. So I've incorporated those as three submodels, each with associated uncertainties. So we acknowledge that the estimates of the number of animals that come out of these types of data have some error associated with them. So the passive acoustic surveys and the visual line transect surveys can inform our estimate of N. The photographic capture recapture can inform our estimate of adult survival. And then we also wanted to know, given what came out of the dynamic energy budget modeling of, maybe calf ratio is something that changes at the onset of disturbance, if we could monitor for calf ratio, if we could collect data on how many calves relative to adults are in the population, would that improve our ability to detect declines? So we added an additional observational submodel here for the number of calves observed relative to the number of adults, which informs the estimate of fecundity. And what we found was first comparing integrated population models versus line transect or PAM data analysis alone. So if you analyzed just the line transect data on its own or just the passive acoustic data on its own, what would your power be to detect trends? It'd be quite low. So this x-axis is the simulated annual decline in the population. The green line is for line transects, and the blue line is for passive acoustic monitoring. So we see that for this species, beaked whales are quite cryptic, they're hard to see, they're deep divers, they're quite difficult to survey visually. So it makes sense that we have quite low power to detect trends over time using visual surveys. Passive acoustic surveys do a bit better, but by combining them and adding in the capture-recapture data, we do much better. And by the time we get to a 1% annual decline, so that's 1% per year decline, we're close to 100% predicted power. Now, the second part of this that I mentioned was we were interested in that calf ratio submodel. So we also compared an integrated population model fit with that submodel and without it. So if you leave out the calf data and only consider the line transect, the passive acoustic, and the capture-recapture, your power is this red line, which again is not bad. But by adding that calf ratio information, we get a massive improvement. So this was an interesting project in that it can guide data collection going forward and how we process data going forward. So what we found was that integrated population models perform better than line transect or passive acoustic monitoring alone, and that the inclusion of information on calf ratio can improve statistical power to detect trends in abundance. But these were simulation studies, and I made a lot of assumptions and made things kind of perfect in some ways. So I think in future, we need more work to make these simulations more realistic and to evaluate whether it is feasible to collect data on calf ratio and what that data would look like. The second example I'm going to give for integrated population models came out of my time as a postdoc here at the University of Washington. And this was a set of projects for Cook Inlet beluga whales. Cook Inlet beluga whales are a geographically isolated population of beluga whales in Cook Inlet, Alaska, that were subjected to commercial and also subsistence hunt in the 1900s, leading to what was ultimately found to be unsustainable hunt in the 1990s. This population has been protected since about the year 2000, so no documented hunting at all, which you would think would give them the ability to rebound. You would think that the population would start to recover, but we haven't seen that from the visual aerial survey data. So the question here was, well, we have other types of data for this population. What if we combined them? We have visual surveys, we have capture-recapture data, and then we also have information about historically how many animals were hunted. So these are from surveys of Native Alaska households who participated in subsistence hunts. And we wondered whether combining these data could improve assessment of this population and help us understand what was going on. The goal was to estimate abundance and trend, and then also life history parameters like survival and fecundity. And then we could ask that question of, well, is survival lower than we would expect, or is fecundity lower than we would expect, et cetera. This is what the model looked like, where we have the population process model again in the middle. This is the population size N. Here it's influenced by these life history parameters that you've seen before, fecundity, survival rates, and then also by this H parameter, which represents removals due to hunting. Again, adult survival is influenced by, or can be informed by capture-recapture data, and the aerial survey data provides information about the total abundance. We also acknowledge that the hunt data were collected imperfectly. They were surveys of households. Not all households were surveyed, and the surveys weren't conducted every year. So we acknowledge that there's some error associated with that. So we estimated there are confidence intervals on this. They're just not showing up. So this is the model estimate of the total population size is this black line here, plotted over the aerial survey estimates of abundance. So as we suspected, the population underwent a steep decline, and then there's maybe a slight increase, but not as much as we would have expected. And we found that juvenile and adult survival were somewhat lower than what we would expect for a population that's just been released from hunting pressure. So after I moved to St. Andrews, a PhD student here, Amanda Warlick, took this project over and moved forward with more complex analyses. So she extended that capture-recapture model to provide information not only about adult survival, but also about survival rates of other age classes and fecundity. So she used whether females were observed with calves in subsequent years to provide information about fecundity and survival of those early age classes. So that's what's shown here. In this case, the total population size is this N here. Again, there's aerial survey data that's feeding into it. And then she had this more complex photographic capture-recapture model called a multi-event model that provided information about various life history traits. In this case, Amanda was interested in the present and the future of Coquille beluga whales. She wasn't as interested in modeling what had happened in the past, so the hunt data isn't included here. And what she did was use the model to project forward in time to evaluate the extinction risk for this population. So this is a time horizon going from 2005 to 2165. Again, there are confidence intervals on these plots. This is the model estimate of the population trajectory over that time period. And then she's plotted the extinction probability here on the other y-axis. So she found that over a 150-year period, there's about a 25% chance that this population is going to go extinct. And she also found through using that more complex capture-recapture observation model that fecundity and non-breeding adult survival were lower than expected. So I think this pair of projects is really cool because it shows how you can use integrated population models in different ways, how you can use the same data set in different ways to answer different questions, and how they can inform our understanding of the past, the present, and the future of populations. In the future for integrated population models, a couple of things that I think would be really interesting to work on. One is extending these models to be spatially and/or temporally continuous. So as I said, when I was describing integrated population models, we're usually kind of cookie cuttering a population and saying we're interested in this population of animals in this specific region. What would be cool would be to extend that to a continuous spatial field. So instead of thinking about a population as being closed and only existing in one space, acknowledging that populations have some distribution and might have different densities in different areas, might have different life history parameters in different areas. These models also tend to operate on a 1-year time step. And it would be interesting to explore whether we could extend that to be spatially continuous or at least seasonally continuous. Think about that. The other area where I think these models will continue to expand is the integration of different data types. For example, environmental DNA. So actually the reason I'm here in Seattle is to work with some folks at the School of the Environment and with Amy on a project to detect marine mammals in the California current ecosystem using environmental DNA. And some of the work that's come out of that, this is work by a postdoc here at SAFS, Tanya Valdivia. This is the coast of Washington. These dots are the locations of water samples for environmental DNA. The red dots represent detections of Pacific white-sided dolphins, and the black dots are non-detections. She then used that data to create a model of the distribution of Pacific white-sided dolphins. So we also have for this species visual data collected from line transect surveys. So it'd be really cool in the future to see if we could build an integrated model that incorporated both of these kinds of, and potentially even more different kinds of data. I'll now transition to talk about close-kin marker capture. Again, I'll go over the basic concept of what close-kin marker capture is, how it works. And I'll give one marine mammal example. So close-kin marker capture has mostly been used in a fisheries context. There are only a few marine mammal examples, and I'm only involved in one project, which is on Pacific walrus. In general, the close-kin marker capture pipeline looks something like this. Ideally, you do a study design and think about how feasible it's going to be to undertake a close-kin marker capture analysis. You then go out and collect your samples. Then there's a ton of work that goes, there's a ton of work that goes into sample collection, I acknowledge, and then also aging and genotyping. So figuring out epigenetic age and figuring out the relationships among individual samples is a massive amount of work. I'm only going to talk about these two ends of this pipeline, but just wanted to acknowledge that it's more difficult than that. Okay, so the basic concept of close-kin marker capture is to extend the traditional capture-recapture framework from recapture of yourself to recapture of relatives. This is a conceptual diagram where we have juveniles are these small fish on the top, and adults are these smaller fish down here. Each juvenile has two parents. Makes sense. But not all adults are parents. Some, like this individual, is the parent of 3 juveniles. This individual doesn't have any offspring. We then go out and sample both the juveniles and adults. So that's what's indicated by these darker individuals are sampled. So we observe some of these relationships, but not all of them. The dashed lines represent relationships that we do not observe. Fundamentally, just the concept is that recaptures of kin are less likely to happen in large populations. So the number of recaptures of kin provides information about abundance. In a little bit more mathy terms, if you compare the genotype of some NJ juvenile samples to some NA adult samples, for each pair of samples, each juvenile adult comparison pair, the probability that any adult, that adult is one of the juvenile's 2 parents is 2 over the population size or the abundance of adults. So that makes sense because you know that each individual has 2 parents, divide that by the total population size. That's your probability that any individual adult will be their parent. So if you do NJ times NA comparisons, so you do pairwise comparisons for every possible pair, and you detect H parent offspring pairs, then you can calculate the abundance, the adult abundance NA, as 2 times the number of comparisons that you did divided by the number of parent offspring pairs that you observed. Now, this is an extremely simplified version of how this works. This is excluding all sorts of assumptions about survival rates and things like that, but just to give you a flavor of sort of conceptually how this process works. The cool thing about close kin marker capture is that it requires sparse, what was been termed sparse sampling. So meaning that your sample size, little n, should be on the order of about square root of your population size of interest, which is convenient when you're thinking about things like fish, whose population size is maybe in the millions. The most common kinship types to use start with mother offspring pairs or parent offspring pairs, but you can also use information about half sibling pairs and even consider self as a kinship type, which I'll show in an example in a minute. So for each pair of individuals, you calculate the probability that they're related. Again, that incorporates what I said before about, each individual has 2 parents, but then you also incorporate information about the probability of survival of each of those individuals. You treat those pairwise comparisons as a series of Bernoulli trials and compare the number of observed kin pairs that you see to the number that you would expect given some population size. Some of the advantages of close kin marker capture are that you can use lethal or non-lethal samples. So because you're not recapturing individuals, it doesn't matter if you kill them in the sampling process. And it also makes it tractable for fisheries because the fish are being harvested anyway. If you can take a tiny sample from each of those, then you can use this method. But you can also use non-lethal samples. Again, you just have to do some accounting for the survival going forward. You don't need to recapture individuals, tractable for large populations. Some of the disadvantage at the moment is that this sort of pipeline tends to be very bespoke in a lot of ways. So for each individual species or population, it requires a lot of thought about exactly what the sampling strategy is going to be, how you're going to do the kin finding and the genotyping. And then the close kin marker capture analysis has to take into account the life history of the population or species, and basically relies on population models. So I'll give an example that I'm currently working on. This is a work in progress led by the USGS and the Fish and Wildlife Service. So walrus are an ice-associated marine mammal that have been hunted for subsistence since time immemorial. They experienced a population decline in the late 20th century, probably because they had reached carrying capacity. And traditional survey methods have provided quite low precision on estimates of abundance. So this is These dots with lines are aerial survey estimates of abundance from 1975 to 2005. And you see that the error bars on that are quite large. Now, this line with the confidence intervals is an integrated population model fit to those data. So that improves things somewhat, but still we could use better precision. I mean, if you think about just, I think it's my, just the scale that they're operating on, Pacific walrus are in in the Bering and Chukchi seas, which are hard to access. They're associated with ice flows, which are mobile, and they're just very difficult to survey. So about 10 years ago, the USGS and Fish and Wildlife Service collected samples, and they wanted to do individual genetic mark recapture. So they went out and biopsied individuals thinking that they would use individual genetic mark recapture. Over a 5-year period, they collected over 8,000 biopsy samples, which is, I just find the scale of that astonishing. And they've now started another round of sampling, another 5 years going forward. Since this project began 10 years ago, technology has come a long way, and close kin is more feasible for walrus now than it was 10 years ago, both in terms of the genotyping and also in terms of epigenetic aging. So the question for this project was, can we kind of convert this, what was initially envisioned as an individual genetic marker capture project, to a close kin marker capture project? And by doing so, can we improve precision in the abundance estimates? Also, since data collection is going to be ongoing for another 4 years, can we provide guidance on sampling, either of which age classes to target or how many samples to collect that could optimize sampling and improve precision of abundance estimates? So this is the distribution of Pacific walrus, is this shaded area in the Bering and Chukchi seas. Each dot is a sampling location from the previous round of biopsy collection. So you see it's just a vast area. And well, I'll talk about this in a minute. You see that some of the samples were previously collected in Russia and some were collected in America. So for this project, we had to consider some things about the life history of walrus. One thing is that they have sex-segregated summer distributions. So only females and calves and juveniles go north of the Bering Strait in summer. So these are almost all samples of females with some juvenile and young males in there. The second consideration is that this species is polygynous. So they form leks where one male might mate with many females, and then some males don't mate at all. And that changes over time. So a male might have one really good year and then never reproduce again. We were worried about movement between US and Russian waters. So there was this question of, is movement, do these animals have a preference? Do they return to their natal site? And given the current state of the political climate between the US and Russia, we're not going to be able to collect samples from Russia in the future. Is that going to be a problem? Finally, they have an interesting multi-year breeding cycle. They're pregnant for 15 months. So what that means is that an individual cannot give birth in back-to-back years. Half siblings, or in a rare case as a full sibling, must be at least 2 years apart, but are more likely to be 3, 4, or more years apart. So the first couple things we dealt with by ignoring males and said, our goal is going to be to estimate the abundance of adult females. We don't care about males. That got rid of the first 2 problems. The third problem, we looked at some tagging data and found that actually individual walruses move back and forth between Russian and US waters quite a bit within years. So there doesn't seem to be any evidence that they have certain preferences for where or for sites that they return to, and no evidence that they pass that on to their offspring. So we could cross that one off. The thing we did still need to deal with was this multi-year breeding cycle. So we basically embedded a hidden Markov model into our population process model, where we said that individuals could be pregnant, they might be in a state where they just had a kid, or they might be in a state where they had a kid 1 or more years ago. And there are different probabilities associated with transitioning between each of these states. The important thing is that they cannot, if they just had a kid, they can't return to this state. They must move here, or they can go back to being pregnant. So this prevents individuals from the possibility of having calves in back-to-back years. We developed a model and a simulation that we could use to test it. This was a stage-structured population model. And as I mentioned before, we incorporated those individual mark recapture estimates. So out of those 8,000 samples, they did get some individuals more than once. We just added that as an extra kinship type and asked, well, what is the probability if I take a sample in 2000 or 2020 and 2021, what's the probability that I get that individual again? So our preliminary results, and I'll stress that these are preliminary, but I did want to share. We're finding that the expected CVs of our estimates of female abundance are less than 15%. And in some cases, so we do years estimates of abundance for each year. In some cases, less than 5%, which is very small. And this is 2 or 3 times lower than what they would have gotten using individual market capture alone. So massive improvement there. And I don't even know how it compares to the CVs of the aerial surveys, but at least 4 or 5 times better, probably. From this, we also expect to get precise estimates of juvenile and adult survival that can inform just conservation and management of the species going forward. In the future, we're going to explore possible changes to the sampling scheme, whether they should collect, what numbers of samples they should collect, and how those might be distributed amongst age classes to optimize this process. And there are also some lethal samples available from subsistence hunts that we would like to incorporate. Again, it's totally possible to have both lethal and non-lethal samples. You just have to account for the fact that once an animal's been killed, it can't go on to reproduce and it can't be captured again. And then we're going to use the simulation to test violations to the model assumption. For example, we've built the model now with no senescence in it. So we assume that females are able to reproduce until they die, but that might not be right. We don't really know for walrus. So we're going to try some simulations where we simulate data where there is senescence and see if that affects our resulting estimates of adult population size. So to conclude and kind of return to some of the themes that I mentioned at the beginning, I really like integrated population models because they make use of existing data and can be adapted to use a variety of different types of data, including data that we don't even know how to collect yet. They can improve inference compared to single stream analyses. And I think using them in a simulation framework is super useful for guiding future data collection and optimizing, basically getting the best bang for your buck. If you have limited resources, what are the best types of data to collect and in what frequencies, and what can you expect to get out of them? For close-kin marker capture, this is a tractable technique. I mean, it's been used in a fisheries context for about 10 years now. For estimating abundance and life history parameters for a wide range of taxa, but there aren't very many marine mammal examples yet. So like I said, I'm just getting into this field, and I'd be really open to ideas or collaborations if people have any thoughts on like a population that you think would be a really good candidate for this kind of modeling. And in the future, as I mentioned before, they tend to be quite bespoke at the moment. So we're also thinking about how we might make this more accessible to a wider range of people so that it doesn't have to be so bespoke for each individual species or population of interest. And then coming back to the overall themes of sort of the areas that I work in, I hope I've shown the value of using existing data and information in models, the potential of new technologies, and the benefits of collaborative approaches. I really value that I get to work with so many different people in my work, and I think it leads to much better science. With that, I would just like to acknowledge all of my collaborators and all of the field biologists and technicians who have collected and processed these amazing data sets. I feel so lucky to get to work on them. I'd also like to acknowledge the Alaska Native communities who have provided information and samples from subsistence harvests. So thank you. Okay, we have a couple minutes for questions. I'm going to let Eiren field them. Okay. Yeah.
I heard you say that, all data is imperfect, some data is more imperfect. When you're doing your IPMs, did you ever look at weighing any of those different data sources?
So this is one of those things that's really common in stock assessment models, but doesn't really happen in integrated population models. And I think it's because in stock assessment, people have expectations for which kinds of data are going to be more reliable, which kinds are more trustworthy. For example, if you have some data that's collected from by a fisheries observer versus a fishery independent survey, you might think that one of those is more reliable. In an integrated population modeling context, we don't tend to have that. So we tend to allow all of the data types to inform the model in an unweighted way. Yeah?
Do you think that environmental DNA could be useful for future improvement in protein?
So the tricky thing about environmental DNA is that it is, okay, so first of all, close-kin marker capture, you have to use nuclear DNA to establish these relationships amongst pairs of individuals. Most environmental DNA sampling focuses on mitochondrial DNA just because it's so much more abundant. The other couple of issues you have is that environmental DNA, you take a water sample, you don't know when or where that DNA was deposited. So you don't know how many individuals it came from is one thing. And then you also don't know. So for example, right now, if you go out and take biopsy samples and you saw a mother and calf pair, you would not sample both of them. You would take just the mother or just the calf because those are not independent samples. If the water's all mixed together and you're just filtering it, you don't know whether, and you get, say, a kinship pair, you don't know if that's because it was a mother-calf pair or because you happen to get, you know, a mother and a calf that had been separated for 10 years. The other thing is aging. So aging is really important for close kin because it allows you to calculate those survival probabilities. And you wouldn't get that from environmental DNA. At least not yet, I don't. No?
So along the lines of the DNA sampling, with the aging and being able to reasonably accurately estimate the relationships between two samples, that's a lot of money.
Sorry, from the environmental DNA or from the biopsies?
From biopsies. It's a lot of money.
Yeah.
Samples doing, I assume you do some kind of reduced representation, but you're not going to do whole genome sequence. So you need, but you still need enough SNPs or something to get accurate data, and then incorporate your probability of getting those accurate. Is this something that's actually being done with those samples? Is there
For walrus, it is.
They're doing all of that?
I believe that the USGS and Fish and Wildlife Service have contracted. We can talk after about the details of this specific project, but if you think about the alternative is to fly a tiny plane over freezing cold water over a vast area. Think about the expense and the risks involved in that. I mean, close kin doesn't seem that unreasonable. So I guess it depends on how important it is.
Yeah, no, I understand. I think you're absolutely right. It makes sense to do it. I'm just curious at how often it's being done, and getting enough samples and enough markers, as well as the age data all together, if that's actually being done in these projects.
So it's being done for a lot of like tuna and sort of large, valuable commercial fisheries. Like I said, for marine mammals, the only other example I know of is Paul Kahn did a bearded seal project. So it's just starting to creep into the marine mammal space. And it's only really appropriate in certain circumstances. Like if you already have a way of surveying your species of population of interest, great. So this is really for a species that we haven't figured out a good way to do it yet. Anything else? Len.
So you talked about close-kin market capture, and you talked about individual market capture, and indeed this was designed for it, so you must get some individual recapture data. So is there some possibility to put the two together or use the individual captures?
Yes. So we've added self-recapture as just another kinship probability to the model. I'm not sure if we've checked I'm not 100% sure on what, I couldn't tell you numerically what it adds. I think it does help, but I'm not sure how much. But yes, there are individual captures, so we use that. Beth?
Yeah, I was curious, you put up the IPM versus stock assessment, and you said IPMs typically do relative abundance and stock assessments do absolute abundance. And being on the IPM side, I was curious because like we think a lot about detection and estimating true abundance from capture recapture models, sort of being able to integrate that in. So I was wondering if you could talk about that difference and how, like, how do you get absolute abundance in a stock assessment, but you can't get it in an IPM?
I'm not saying you can't get it. I'm saying sometimes the goal isn't necessarily to estimate absolute abundance. Sometimes people are more interested in the trend for conservation ecology purposes. Maybe you're more interested in the trend over time than you are in the absolute abundance. To come to the capture recapture question, I think historically, so capture recapture data can be used to estimate survival. It can also be used to estimate total population size. Historically, there was concern that basically each of the data sets fed into an IPM needed to be independent. And so people didn't want to, quote unquote, reuse the capture recapture data to inform both the estimates of survival and the estimates of abundance. There was a simulation paper a couple of years ago that showed that maybe that's not an issue. So maybe we could do that for going forward. And yeah, I think if we can, then great.